bell work type stuff. There's a number of ways you can open up with it, like they were talking about the vocabulary. I guess making people kind of letting them think about it a little more, where they just come from English or some type of something where they were reading anyways. But what we're talking here is another thing we can do is we can ask them what features they have. Give them examples like hair color, eye color, maybe even put, that they have to put a certain amount of traits on there just so they don't say, all right, I have brown hair, cool, I'm done. And then our hook here is, because we're talking about genetic diversity, we ask them what would happen if we all looked alike, if we all had the same genetic makeup, genotypes. We could throw in vocabulary words for them. And we give them this brown eyes, brown hair, fair skin, etc. We use some of their traits.
When we get to the electron transport chain, we sing the little Aretha Franklin part. Change. So my kids walk around school singing that, but it helps them to remember that that's the part where it goes from the excited electrons into the valence cycle.
Then we had multiple representation, and Ashley came up with a table and a story correlation with that table, if I'm correct. Mm-hmm. And discovery learning, film clip review, discussion, and then our assessment, and we will evaluate the tests, discuss, and then retest it needed.
The same lady that did the play for photosynthesis, and she has somebody dressed up as Granny Granum, and then she has somebody that carries a knapsack because they're in ADP, and that makes an ADPH, and she passes out water to fill the holes, and she passes out sugar packets at the end to go through all that stuff. We asked her about how do you cover cellular respiration in class, because the kids kind of have a hole with that as well. And she said that when you get to the end of photosynthesis, you have one sugar, and that's 2 PGALs, and she calls them pretty gals. And these 2 pretty gals then at that point fall in love with one man, Calvin. And so they fight over Calvin, and the one lady that wins Calvin, of course, then eats crabs all the time because that's the Krebs cycle, and they own a transport company because that's the electron transport chain. They live by the water because that's what they make at the end, and they have lots of money, which is the ATP. But the poor little pretty gal that doesn't run in with Calvin, she turns to alcohol. The alcoholic fermentation. So I do that little story, but again, I have all this illustrated out on the page. But they get it. I mean, they sing and they're,
You got to describe the process of meiosis. You have to cover these points. That you're talking about going from a diploid to a haploid. It occurs in the gonads, producing 4 cells, again, haploid cells. 2 divisions rather than 1 division of meiosis. First cell division is where the homologous pairs get split up, and then the second cell division is when the sister chromatids of the chromosomes get broken up into the haploid cells. Hands-on activities, there's a myriad of activities. You can do like the one we did here with clay, with beads, with kits from various chemicals or educational supply companies. Gizmos has some nice activities with this. There are also lots of online computer activities if you have computers. A lot of our schools don't have computers in the classroom, so it's a trip to the media center, hopefully. Anyways, and what else would you like me to talk about? That's it. I mean, obviously, at the end of the day, you summarize, and again, always we want to go back and touch back and emphasize the difference between meiosis and mitosis.
And so for our objective that we had, it was explain the interrelated nature of photosynthesis and cellular respiration. We tried to do a 5E model for this one. So our engagement would actually be setting up a lab. So we would have the students use bromothiamine blue, blow into them, change the color from blue to green or yellow, then pour that water into a test tube that has some type of aquatic plant. After that, we go into exploration when we basically just talk to them and see what they thought was going to happen. Okay, hopefully they get the right answer. Then we go through an explanation about 10 minutes of an explanation with a PowerPoint. And then the last thing would be elaboration. And there's kind of 2 things you could do with that. You could do like a gizmo, which the one in for this section actually is pretty quick, so you might be able to fit it in. Or you could do more of a student engaging activity with the skits or songs or a cartoon, a drawing of some sort, and have them basically explain the objective with that explanation that you just gave them 10 minutes before. And then wrapped up in that elaboration, you do your evaluation where the teacher is basically checking. You can also have the students check themselves and make sure that other students and other groups are actually getting the right information. So
we thought that we could start as a hook with a clip from that show Little People Big World. Then like gets kids into like, you know, the reality TV craze, whatever. But it showcases achondroplasia. And so we could talk about how we have two people with achondroplasia as the parents, and then they have children who are normal stature. So you could kind of discuss with kids, how does that work, how does that happen, looking at their, maybe we could do some kind of stories and stuff, looking at their genetics. I don't know if anybody else like who does biology has heard of like the Making Babies lab where you're flipping pennies, you know, determining what genotype they get. So you have your kids make babies in class, and then they get to see what the how the genotype affects the phenotype and blah blah blah. And then we have karyotype labs too, where you can give them homologous chromosomes and put it up and kind of see again, you know. And this is reinforcing meiosis and sexual reproduction.
There's some big gizmo labs also for the blue chickens. And some really good ones. And also there's a big karyotyping lab as well.